Yes, we can.
Perfect. All right. Thank you, Liang and Dr. Liang and Chun Hong and then Yang Xi and thank you for CBA for hosting this important session. So very timely. Just to put the progress in the perspective. So maybe many of you already know, and it took us 13 years and almost 3 billion to sequence the first human genome, right? So back in 20 years ago or 25 years ago. So today, so the same process can be done in about maybe less than 24 hours, and under $100,000, right? So it's still evolving. And so let's think about the speed of the transformation in biomedicine. So today, in a computational pathology, is at the same kind of inflection point. So the question is no longer if the computer pathology will impact the patient care, but how fast, right? We can responsibly integrate it into the clinical practice. So that's the journey we're beginning exploring today. Again, my name is Tong, and thank you for the introduction. But before we start, so let's do a quick polling. Well, before I needed to do the polling. Oh, here. All right. So um, just trying to understand um, what our audience the background. So maybe you're in mainly focused on the clinical and mainly focused on the research, or if you're experts in the data science or AI, or you're one of the pharma or biotech, representative, right? So let's see how our audience are, so we're looking, and then so we can probably tailored how we can probably discuss the next session. And then how you familiar, the second question is how you familiar are in the computer pathology. And so you're kind of a hands-on person or you just heard about it, never heard about it. And so, yeah, so give a sense about what's familiarity of your expertise in the computer pathology. And the third one is how to best describe your current role, right? So research in academia, in a hospital setting, or your pharma, and I think this is a repeat, or you're working in the tech company and working on the AI driven or others, right? So let's say here, yeah, significance in pharma and pathology it's a very even, I would say, right? And the first question, and then the second question, yeah, it's cross them, different familiarity, right? So yeah, it's very interesting. And the last, I think is yeah, so it's pretty much almost half of you are from the academia or research. Okay, that's great. That's great. So I'll give just one, or maybe 5 seconds more, and then we can move on. All right, perfect. I'll end the poll. So before we start, and so this is my disclaimer, and so I think the presentation is intended to provide educational information, and it will not discuss or promote any investigational or approved drugs. So also, and I'm a currently employee of AstraZeneca, as I hold the shares in the company. So this is my disclosure. All right. So what are we going to talk about today, right? So going to this slides, and so over the past maybe 20, 30 years, and so the biomarker testing has expanded rapidly from the single endotype assays to multi-layer the panels, right? So we now routinely interrogate the 4 classes like showed on the slides, mutation and fusion and the copy numbers and the protein expression, right? So different target demands different tools, right? So that's why we have several different technologies are working on, focusing on the different areas. So let's say the DNA RNA sequencing detect the mutation and fusion, especially RNA sequencing can detect the fusions. And the NGS the next-gen sequencing to consolidate this into a more broad or comprehensive panels can address some of the multiple genes or actional genes. And then the fish, can or ish can do the fusions, and then the CGH can expand the genome-wise, right? So in the moving to the right, and then for the IHC is not something and new, right? It's been there for maybe 50, 60 years, since 70s, so started routinely used in the clinics. So it's not a new technology, it's old technology. In how this is still being utilized in the clinical today, right? Because you all heard about probably the PDL1. That is a very very famous expression biomarker, but also the others like ALK, ROS1, and the C3, CDAs, to look at the microenvironment, the tumor microenvironment. Also, some of the new ones like TROP2, HER2, HER3, right? So those are using the IHCs, especially the TROP2 and HER3. So those are also still using the IHC, but the IHC is the old technology, right? So thinking about this is IHC is a pathology looking or staining the slides and looking under microscope and to see, okay, this is percentage of positivity, right? So it's 50% of positivity or 25% of positivity. So that's kind of a thing we do. We get the number, we got the positivity of the different protein expression biomarker. But it's one of the big challenges that is often we heard some people saying, okay, this is not accurate and this is so subjective, and it's how we can make this more objective. So that come to our today's topic is the digital pathology and the computational pathology. So can make the IHC, the image-based technologies more or upgraded put it into the way. All right, let's move to the next slides here. So the pathology, and no matter it's the conventional or the digital pathology. So you see from the left side, and so the tissue acquisition and preparation and the HN slides, those are the same, right? So no matter you're using conventional pathology or you're using digital pathology, they're the same procedure they have to follow. So since probably half of us audience are from academia and the research, you may not know exactly how this tissue handling works in the clinical lab or in the pathology lab. So anatomical pathology lab. So basically, the surgeon or the tissue takers. So when patients are suspected to be cancer patients, right? So you got to get the cancer tissue and to identify this is a real malignant cancer cells, right? So that is the tissue you take from the patients, but you have to prepare the tissue, and on the slides for pathology to look at is a malignant, is a cancer cell or not? What's the histotype, right? Histology type. And it's adenocarcinoma or others. So those kind of things are the same procedure. But for conventional pathology, it was basically just pathologies using the microscope to manually check and the intensity of the staining and under the microscope. Right, so that's the manual assessment. Then I have a manual report, and so all the slides will stored in somewhere for long term because that is SFP sample fixed in the room temperature can store for years in years. And so moving to the digital pathology is just one additional layer of the conventional pathology. Instead of using the manual assessment, so they'll using the scanners, scan all the slides to our digital format. And then the digital format is very, very high resolution. So the pathologists can look at the images through the monitor instead of using the microscope. Right, so that is the key difference between the conventional and digital pathology. So what we can consider conventional is an analog, but digital is digital, right? So that's the key difference. And then so computational pathology is on top of a digital pathology because AI-driven this assessment needed to have the slides to be digitized first. So that is a prerequisite for computation pathology. So on top of or based on the digital pathology, the AI will use algorithm to assess or to trying to give the score of the different biomarkers instead of a pathology manually, or even they're checking the digital pathology, checking the monitor, but it's a manual process say this is a positivity, how much, right? But computational pathology is using the AI-based algorithm to do that, to analyze the whole slide imaging, right? And then after that, the pathology have some QC and to confirm that diagnosis or certain of the subtypes, and then go into the digital report and digital storage. So that's the key difference between the different conventional pathology, the pathology, and the computational pathology. So talking about the digital computational impact, impacted the pathology overall, it was from the different angles, from the diagnosis, from screening, from prognosis, and quantitative assessment, and then now it's more potential predictive biomarker, right? So for treatment decisions. So you can see here's a list, and so the different at a different stage for different purpose. There's also already have quite a bit of the companies or AI companies or startups are working on the different angles. And so I think the next slides we'll talk about is or next couple slides we'll talk about this timeline and which companies have which of the milestones. But overall, there's many of AI-driven companies working on different biomarkers for different purposes at a different stages. All right, so this is kind of a timeline with some of the key milestones. It's not exclusive, right? So I didn't include everything, but this is kind of just some of the key milestones. The blue color is more digital pathology. It's a digital technology. And the yellow or yellowish color is more from the computational, like a software based. So you can see there's a cross from the last 20 years pretty much, or more advanced, right? But back in if talking about terminology or the first thing to do, the machine learning start, let's go back to like 70, 75 years ago. All right, so and just into this year, the first 6 months, and then you see here, there's substantial the key the milestones, right? And so this is not all. I think so I just put the three of them here, but there's another thing. I think just right after. I think it's probably in July. There's another company called Ultra AI, which is for prostate prognosis, have a breakthrough designation by FDA. And in August, there's a big news in the digital pathology and the computation pathology new award is Tempus acquired Page. The Page is the company, it's the AI companies. So they have their first clearance by FDA back in the 2021. So you see, there's kind of a different approach, different milestone, but the Tempus acquired Page is combining Tempus of the genomic data, EHR data, the spectral data, plus imaging data together, right? That's a multi-modal approach. That is the future direction, so where we're going. So this slides may all data already, because this field is evolved so fast, and some of the common scanners, I would say worldwide, but it's not the real case in China, because China has some of the local scanners too, which is cheaper. So I'll pause here. Let's go back to the second poll, and let's try to have some of the guests. So how many pathology labs there in US? Just a well guess, right? So take a look or take a guess. All right. The first question is how many pathology lab in US? The second question is what percentage of US lab are digitized for whole slice imaging, right? So with the scan, one of those scanners. And the third question is roughly how much does it cost to set up a mid-sized digital pathology lab? So that including not just scanner. So if scanner just one step, just one thing, but you have to consider storage, IT, and infrastructure plus the experts, right? The FTEs, right? Everything included. So give a wild guess. Okay, I see. They're about 40% of audience guess about 5,000 pathology lab in the US. So based on the 2023 data, it was roughly 9,000. And so that is so how many of the pathology labs we're talking about. So it's not realistic to digitize every single lab. That is the one thing because the throughput, because a lot of things are different factors. And then the second question here, what the percentage of US labs are digitized for whole slice imaging? I think a large portion, like 40% is saying about 10%, and 27% is saying lower than 5%, and another 20 something saying 25%. It's actually a little bit slightly lower than 10% at the current stage. The third question is roughly how much it does cost to set up a mid-sized digital pathology lab? Well, I guess majority of them are guys are very right. It's a $2 or $3 million. So about 30% have this answer. So it can be more or less, right? Depends. But I think that roughly the ballpark is 2 or 3 millions. And so because you have to have your scanner, you have to have the storage. So why we're thinking about storage IT is very important is because each of the slides, the whole image slides, if it's not compressed, the original image can be as big as like 30 something gigabytes. But most of the slides already been compressed after the imaging processing. So it will be roughly to 1 to 5 gigabytes per slice. And so if you think like Memorial Sloan Kettering, they have over 10,000 slides per year to scan. If you think of that to scale and plus 1 gig and how much space they need to maintain, that is just 1 year, right? So if you think about the storage of the digital image for 10 years, that's how much space you're going to have, right? So that is how to maintain, and those are all the costs. Plus, the data, big data, it will transfer through your network to cloud because you don't have enough in your local storage. You have to upload that to a cloud, right? So those are the bandwidths, your infrastructure. It totally changed that. So it's all impact, the overall infrastructure and plus the FTE. So that's why the cost, the upfront cost is very high. And it could be lower. It could be lower. And so, for example, the scanner could be lower. And so maybe from, 500K or to 200K or 300K depends on, so which company or which area. So like China, some of the, manufacturers have a fairly low, the price point compared to some of the US based. So those are some of the key challenges talking about it. So that's from the hardware, from the scanning, from the infrastructure. All right, so I'll share the slide. I'll share the poll for you to take a look, and so we'll move on to the next slide. So why we need this digital pathology or computational pathology, right, to help us. But this is one of the papers and published. And so it will use these two pictures to compare. So if you take a well guess, see, okay, using your human eye to see the A and B, which is more intense, right? So most of us probably say A is more intense than B, but in fact, it was equal, right? So that is where it can misleading you because of some of the other objects in your environment for human eyes. So that's why we need data pathology and computational pathology, right? So comparing the digital pathology and then traditional the HC pathology, the score for the scoring purpose, and you see here is some of the key differences: quantitation, right? The quantitation, which the traditional one is not are fully quantitated, right? So because, for example, we use the PDL1 as the example. The PDL1 can express from 1% to 100%, right? So it's a continuous score. It's not the, kind of the cutoff. You can use the cutoff as a standard, but the expression level is a continuous, right? So you get to give 10%, 20%, or 25%, or 50%, right? So that is the data which only semi-quantitated by the human or by pathology, this manual scoring, but can be easily adapted by the computation pathology to give a continuous scoring. Another is a dynamic range, right? So especially for the low level staining. For example, so right now, and some of the drugs need probably the HER2 testing, but the HER2, potentially HER2 low or ultra low, right? So in a normal staining or it's a strong staining, it's easy for human eye to capture or to know the percentage. But it's going to very faint, very low level of the staining. So it would be really, really hard for human eye to capture everything, right? So that is where the computation pathology can contribute more. So that is the second, and then spectral heterogeneity, right? So that is a cutoff based on in quantity based on the per cell the distribution and the distance across the entire slice. So those are some of the key differences between the manual and then digital or computational pathology. Talking about AI, right? So everyone knows the buzzword AI, but what is AI, right? So AI is a very, very broad word, right? So it can do a lot of different things. So this is where we just lay out artificial intelligence. That is the broadest word. Is something is just get a computer to simulate the human intelligence. And so down below, that is a smaller scope called machine learning, right? So you got to have a machine learning. So I don't know how many of you computer science background. Back, 20 years ago, when I had my computer, study. So back then, there's not talking about, the buzzword AI, but I think the, concept is similar or same. So they're talking about data management, data clustering or classification or so it's trained or not trained, so supervised, unsupervised. Those concepts are the same. So machine learning are doing the similar thing. So kind of training the data, and so getting the algorithm better and better, right? But until the deep learning, until the smallest circle, and so we are not talking about the real AI for computational pathology because the computational pathology will need to use is a more advanced model to predict or even self-train algorithm to adjust. If you're talking about like NGS bioinformatics back in the days, right? And people are building the algorithms and doing a data analysis. And so if you fine tune those data point, the parameters, you need to manually to change the parameters. So now the AI can automatically adjust those parameters to better fit for the best outcome, right? So that is the difference between previous machine learning or artificial intelligence with the deep learning. So this is just one example of the HDN slides, and just the from the left is the whole slides, but on the right side is the zoom in, so with the cancer cells. So this is the picture, um, whole slice imaging picture I borrowed from the pastpresenter.com. So the credit to them. All right, let me just stop sharing this poll result to without interfere or present. Okay. So how this deep learning works, right? In general, it was just a lot of people, if you don't understand the AI or how the deep learning works, it's a black box to you, right? So inside of the black box, it's just different layers, hidden layers, right? How it works. But so put it a simple way, right? So is the machine reading the slides and then converted it to the digital, and then from the input, the layer, it will take the features. So the computer and it read the slides as a feature, so intensity. So they will be, like a vector or anything. So they and also they put into the translate into the hidden layers for different process. It depends on different model. So that is overall the flow. But most of the common deep learning models for whole slice imaging analysis today are, I just kind of listed here. The very top one is called convolutional neural network, which is, often people say that CNN, right? If you see CNN in the computation or AI world, it's not a news agency. It is the model used for whole slice imaging analysis. So I kind of a list here. This is just based on, may not be, very accurate. I just based on my personal kind of experience and putting this together. So some of them may be, outdated already. So because this field is involved too quick. But overall, you see the convolutional neural network is a fully trained model. And then they'll have MIL, which is the multiple instant learning, and we'll have a vision transformer. We have graph neural networks, and at the very bottom is a foundation model. And then that is, I feel this is the future foundation model, but there's definitely for different purpose, for different applications. But going back to CNN, right? So this is the most mature model because they start early. It's a fully trained and fully supervised model. And then so how that CNN works, right? So it's a convolution layer and a polling layer step by step. I think the next slides will slightly better to explain. So for this convolution, that is, you take a filter or kernel, which is 3 by 3 in the middle, right? As your parameter, it could be 2 by 2, or it could be larger. So it have a different advantage or a disadvantage of smaller or bigger. But the concept is the same: is you using that kernel as your filter and apply from your whole slice corner by corner or in the 1 by 1, right? So moving. So how that works is you using, if you're looking at the right side, so it's a 13 times 1, which is all this corner times this corner. That is how much you get, right? Plus, oops, sorry, plus 20 times 0. I don't know why it's moving. Sorry. And then 15 times -1. So just, go by everything, time all 9 of them, and then add up is a 12. That is our first output feature. So why we're going to this? It's trying to simplify the high resolution image. Trying to have a much more feature, and to simplify, have a smaller feature, which is still representing the data, right? So because if you have too much data and you need so much, powerful computing resource to calculate, that is, too expensive. We can't afford it. So that's why some of the model trying to, simplify, retract some of the features, the key features from all the different small features, and putting them together. So you can just shrink and shrink and shrink, by layer and layer until it got to a size is easily can handled by the computing, right? So that is a concept. And after that, convolution step, it will be the pooling. So what is a pooling means is here, you get 2x2 kernel site, and you can also identify, define a different size, right? So I just using 2x2 as an example. And the stride, is moving is by one feature or by two features, right? So this is where, 2x2 kernel size and stride by two. So, you can see here, it's called maximum pooling. It could be average, it could be different, but, for here, as an example, I'm using maximum pooling, which is the most common one. And then so you pull the 4 digits from those four, and the 7 is the biggest one, right? I put this 7 into here, and then next is 9 and a 4 and 8, right? So that is my pooling. So you see here is from previous the whole slides, and which have a bigger matrix, and then down to this 4 matrix. This is the concept to get all the features extract from the original imaging or the input layer, and then simplify, still contain the most critical elements. Right. With that, I think this is kind of just one of them because the CNN. But remember, a couple slides earlier, I talked about this several different models, AI models work on the same analysis. What's their advantage or disadvantage? I think probably it's easier. I put this way. And so you see, this is construction work, right? So what they're focused on is a focus on just the bricks. So when they're part of the brick by brick, it just focus on that particular brick. It's a leveled. It is equal with the others, right? So the whole focus is on those small area. So that is how exactly the CNN works. The CNN is focused on the small square or small feature size. And then it goes through all the small features until the whole slice is scanned. And then moving to the second, that is, we probably can think about this inspector, so the house inspector, or maybe designer, or I would say probably is project manager. Let's say put it project manager. So the project manager will inspect the quality of the construction without looking into each particular bricks, but it will oversee the whole house or the whole room, right? So how that build is leveled overall, and is there any of defect, any of the, so it's from that angle. So that is the, kind of from the higher level, that's the whole slide level, right? That's a different the algorithm. So focus on this is the house inspector, right? So the house inspector, and it just checking. So when the house is built, it will just check the overall quality. It's not going to see brick by brick. It's the whole slice, right? The checking. So that's a different approach by different models. And then if you go into another one is, so this is we call GAN model, which is kind of you have, well, I think the picture here is doesn't show is that clear, but if you're thinking about as a someone trying to print a fake money, right? So you print the fake money and you're trying to spend it, right? So there's the cops trying to catch you because, well, they have to inspect the money is real or fake, right? And so with the time, it kind of played a game between those two. So the criminal, it will get better and better, right? So those, you're easily, hard to find that money will be fake. But the inspector or the cops were trying to increase their level to identify this is the real money or fake ones, right? So that's kind of a GN model. And also we have a foundation model, which is kind of, it's already pre-built a lot of different components. It's just putting together, right? So that's a whole big construction company. So this is kind of a different approach by different model. I think this is easier to explain since a lot of us are researchers and not have too much of a computer background. So I just don't want to go into the too details and lose your interest. So overall, this is a kind of a, so different thing, different and players, just like, or different models, just like different players here, as we just mentioned about it. So now going back to clinical lab, or I would say not clinical lab, it's an anatomical pathology lab, right? So once you get the biopsy and you are fixation and you're embedded, and then you have a FFP sample ready, and then put it on the slides, stain, which and the Z staining, and then your diagnosis, right? So this is the key part. I think earlier slides we already mentioned the first three steps are the same, no matter it's a conventional or digital pathology. So now you're moving right, that is where consider or you can do different things. You can do IHC staining or other downstream, molecular biomarker testing. So that you go for IHC, you go conventional or you go digital, and then so that's a different route. But at the end, so you are going to get to our fully pathology report. And then so now, besides IHC staining, I think as earlier slides also show, is there's another, some of the companies are going early, is going to scan the H&E slides using H&E to predict the mutation, using H&E to predict about EGFR mutation or predict about HER2 mutation, BRCA mutation. There's quite a few, or MSI, so some of them. And so they are, doing this currently. It's still under development. Some of the data show is very promising. The others still need further development. But this kind of a directions where the AI-driven computational pathology, how they goes. All right, so now going back to QC side, because a lot of different models running, and so how can I trust? I think the biggest question. So when I talk to pathologists at a conference or visit their labs, and so the very first question is, how can I trust? How can I rely on this data? I don't know, right? So I'm not comfortable to using digital pathology or computed pathology if I don't know it, I don't understand it, right? So knowing that is because there's a lot of different variables here across the pre-analytical, analytical, clinical validation, where so others, there's many steps to validate each of the models, and so you get to show and all the steps and provide the high confidence to the clinicians or pathologists so they can adapt it, this kind of approach. So going to the clinical, this is one slide from the one publications talking about what if the tissue on the slice is cut, the slice is too thin, or you have a folded, if you have a bubble, if you have any of the other artifact, how the AI handle that, right? So, or another case is, well, when I have our patient samples data or collected in the one hospital, for example, I use Memorial Sloan Kettering again, and they have thousands or maybe millions of data points they can use to train their algorithm. However, that is only their one institution. What if and something different? For example, the staining procedure or pre-handling, right? So there's a lot of variables. One lab may not be same as the other lab. If you're training your model only based on the single institution, so your data is, or your model is not going to predict while for the other institution's data, right? So that is something and we call domain shifting or any, so we have to have some of a standardized or normalize a procedure for the AI to generalize and all the different parameters, and they can train them ourselves and fill in the best parameter for them. But so this is a kind of a one example saying what kind of artifact or some of the low quality slides AI can handle. So key challenges. I think I think throughout the talk, I already talking many of them. So how I trust data. So that is the clinical adoption. The very big barriers. But also a very big one is the upfront cost, the investment, right? So, some of the academic centers probably not that sensitive to the cost because they have the research funding, they have some of their funding resources they can support. But for some of community-based hospital, they are, very sensitive to cost. And so if they cannot get reimbursed, why we're going to invest this much for nothing, right? So we don't see the value, right? So you got to have the economic value to bring to them to see why you need to use the AI or using the digital pathology to help you. It's not going to replace you, but it will be help you to simplify and more accurate diagnosis. So there's a lot of different, perspective for the key challenges. I'm not going to read it one by one, but this is kind of just overall and the situation about a few of the key challenges that we still need to address for computational pathology in the clinical. I will skip that. So since Cheng's talk will mainly focus on regulatory anything, so I'll leave this to Cheng and skip this perspective and then moving to the future application. So currently, I think I already introduced some of the models or how the clinical applications are working on. So what the direction we're going with the AI computational pathology. But there's many other things we haven't talked about is, for example, digital pathology. So the one big benefit is telepathology, right? So you can have a second opinion. So if you have your slides, you're not sure this is a diagnosis, you can easily share the digital image to some of your colleagues or remotely, even from one country to the other country if you want. So it's much easier for the global health and so for second opinion, and also because of the and the AI evolving, and so now is more and more clinical trial drug development are utilizing the AI to apply the AI technique into the drug development cycle. Right, so from the screening into clinical trial, how to recruiting patients using AI models, better recruit and quicker, recruit, and two, the data safety, you're using the AI to monitoring. The whole drug development plan can be applied by AI technology. So it's where it's going to be more clinical, relevant, right? So, some of them maybe go a little bit faster than the others, the pace. But overall, I think the technology or the future application was very bright. And so I think the multimodal computation pathology definitely is a future. So we're integrating the imaging, so CT, X-ray, the radiology, and plus the omics data and others modalities together with the pathology images, right? So everything's combined to predict the patient. It maybe predict the patient's response to certain drugs, certain type of drug, maybe predict the patient prognosis, maybe monitoring the patients on treatment. So there's several applications can use this model to help. And before I talk about is a more fully supervised
Hi,
model,
Tong.
which is CNN. Yeah,
Tong,
sorry.
hi. I'm so sorry to interrupt you, but we have a limited time and we
Okay.
scheduled to conclude at 9:30 and we have one more presentation and the panel discussion. We do it's a very interesting topic, but I'm so sorry for interrupting.
I'm sorry. Yeah, I'm
Oh,
sorry.
no
Yeah, I
worries at
didn't pay
all.
attention about this. But yeah, I think I'm, this is the final slide to conclude. And so now I think I'll get back to our moderator. Thank you.
Thank you, Dr. Yin, for this educational and insightful presentations. I've noticed that some of the audience are eager to ask questions, but as a housekeeping note, we will save the Q&A and panel discussion towards the end of this webinar. So please, but we encourage all the audience to type in your questions into the Q&A box, and we will get to that at the end of this webinar. Now let me to introduce the second speaker of tonight, Dr. Cheng Cui. Dr. Cui is a seasoned regulatory affairs expert with over 10 years of experience in both health authority, the US FDA, and the industry, particularly in the areas of digital health technologies, biomarkers, and the in vitro diagnostic devices for cancer diagnostics in drug development. Dr. Cui spent over 6 years at the US FDA, where he served as a software reviewer for artificial intelligence, machine learning, SAMDs, lead reviewers for digital pathology and computational pathology devices, and in his last years as a post-market team leader. From November 2020 to March 2022, Dr. Cui joined Janssen, where he served as a regulatory affairs director for the use of digital health technology in drug trials. Since April 2022, Dr. Cui joined AstraZeneca as the Senior Translational Regulatory Affairs Director at AstraZeneca. He has been playing a critical role for the development and the implementation of computational pathology innovations in drug trials to enable precision medicine. The title of his talk today is From Pixels to Patient Selection, Regulatory Perspective on Computational Pathology in Targeted Therapies. His presentation will highlight the practical solutions for navigating the regulatory frameworks, challenges, and opportunities in AI-enabled computational pathology. Let's welcome Dr. Cui to this virtual podium. Dr.
All right. Thank you so much, Eric, for the kind introduction. And good morning and good evening, depending where you are. My name is Cheng Cui, and I'm the Senior Director of Translational Regulatory Affairs at AstraZeneca. And today, I'm very glad to provide some perspectives of mine on the use of computational pathology in targeted oncology therapy. All right. This is my disclosure and disclaimer. And for today's presentation, I will provide 5 use cases for the use of computational pathology in targeted therapy, along with the corresponding regulatory considerations for each use case. And following that, I will share some practical solutions to navigate the regulatory challenges. And now let's take a look at these use cases for the use of AI-enabled computational pathology in targeted therapy. So computational pathology can be used in many ways throughout the whole drug development pipeline and also in the commercialization. I will just name a few use cases, starting with the pre-screening tool that is used during patient enrollment to enrich for genomic biomarker with low prevalence. After that, I will introduce for the use of computational pathology in endpoint assessment or to aid the pathologist in making a diagnosis. And it can also be used for cancer prognosis. And even though it is not for targeted therapy per se, but I thought it's worthwhile to mention here as a use case. And finally, computational pathology could also be used in companion diagnostic devices to direct patient treatment by assessing their biomarker status. So first of all, let's take a look at the pre-screening tool use case. So HIE staining is already done on almost every cancer patient. And it is the very first step to tell the pathologist whether we are dealing with cancer or the cancer is, or there is no cancer. In addition, it can provide the tissue architecture. Therefore, the HIE stain slots are typically available for almost all patients. And the recent research has already uncovered that certain histological patterns on the HIE stain slides, they may correlate with specific genomic changes. For example, FGFR3 mutations or germline BRCA mutations. And these mutations are rare, but they are critical for precision therapy and also clinical trial eligibility. And people have trained deep learning models on thousands of HIE stained slide images, and then to correlate with known genomic mutation status. In this way, the algorithm can learn to predict the likelihood of a mutation directly from the slides without any additional lab work. And instead of sending every patient for a costly central molecular test, as a pre-screening tool, we can first run the AI on the existing slide. Only those flagged as high risk can then be sent for formal molecular screening. And low risk cases, they will be skipped, and then the slides and samples can be used for other purposes. Let's say if when the pre-screening tool is optimized for high sensitivity, for example, if the model can achieve 97% of sensitivity, that means, well, the tool can catch 97% of the true mutation carriers. And if the same tool can achieve specificity of 50%, then that translates to a 50% true negative rate, so that they can be avoided for the molecular testing that will really save the cost, and also the patient can be directed to another option. So such a pre-screening tool can boost the trial enrollment, can ensure patients receive the right genomic testing when it truly matters, and also cut unnecessary laboratory cost. And the precious patient samples can be prioritized for other care options. And for such pre-screening tools, FDA has already provided very clear guidance in their guidance document. So I have the reference down at the bottom of the slide. And later, the FDA consider when using those test results to make recommendation about participation in marker-driven therapeutic product clinical trials. It is a process that is essentially pre-screening subject for eligibility. Then the pre-screening can create particular problems. For example, for sponsors attempting to evaluate a novel therapeutic product for the safety and efficacy in an intended patient population, and also for the IVD manufacturer attempting to provide an unbiased demonstration of performance of IVD product. And then the pre-screening can result in a biased clinical trial population that does not represent the intended use patient population. And there are certain mitigations provided in the FDA guidance document. So for example, one approach to test for all samples, that means not only the patient who were forwarded for central testing, then there's a certain level of negative patients. They will be still need to be tested if not all patients are tested. And then, of course, in the cases when the pre-screening is unavoidable, sponsors should be aware of the potential for bias and evaluate whether the expected prevalence of the biomarker is being skewed by the pre-screening procedure, and develop appropriate approaches to adequately address potential selection biases. Okay, so maybe I will just skip this slide. And then the next use case is regarding the endpoint assessment by using the computational pathology to automatically assess the pathological complete response. So the PCR is increasingly used as a surrogate endpoint, particularly in the breast cancer for regulatory approval decisions. And but the traditional method, normally the pathologist will review the HIE slides, is inherently subjective, and there is a great level of inter-observer variability, and that can shift endpoint status, especially in large multi-center trials And that shift can potentially skew the data. And there are already published literature where an automated AI-enabled algorithm that can A, flag a case as a PCR, and B, when the residual disease remains, it can quantify it precisely. Okay. And essentially, the deep learning model can learn to recognize the tumor architecture from the H&E stained images and also the immune infiltrates, then assign the probability for complete response. And then for non-PCR cases, still have residual disease, it can provide an estimate for the remaining cancer cells. So as you can see, by using the algorithm, it can provide an objective review, and also the review could be quantitative. It can also be very reproducible, and also can enhance efficiency during the trial. But normally such tool is used only during the clinical trial. So there are certain regulatory considerations for that case. And then of course, yes.
Sorry to interrupt you. So one audience mentioned that your voice has some echo. I'm not sure if you can like switch your speaker a little bit.
Okay. All right. So let me just try another way. Just a moment.
All right. Thank you. We cannot hear you now.
Okay, can you hear me now?
Yes.
We
Okay,
can hear
great.
you now. It's
Better.
clear. Thank you.
Yeah.
So let me reshare the screen and then we can pick up from where it belongs.
All right. Thank you.
Sure, no problem. Thank you for letting me know. Okay, let's do it one more time. Okay. All right, so let's continue. So when we are using this tool as endpoint assessment during the trial, so the sponsor need to realize and also have a good estimate of the potential bias in the training data that can lead to inaccurate assessment for certain patient subgroups or for certain tissue histologies. And the FDA may require several things. I will just go through a list very quickly. First, they may require a study demonstrating the performance. And also FDA may require the review of the validation report. They may also require documentation on training and tuning and test of the AI model. And they may consider the tool for investigational use. That means FDA may require for the development, also validation of the tool to be put under medical device design control requirements. And I think it's very important to align with the FDA before this tool is implemented in a clinical setting. The next use case is for the computational pathology to aid the pathologist in making diagnosis. So there are already a few cases. Of course, as you can see, they can be used only to aid the pathologist, but not to make the algorithm itself is not making final diagnosis. It's still the pathologist make the final diagnosis. There are also apparent benefits. It can improve the accuracy, improve the consistency, and also improve potentially the efficiency. And for such devices to aid the pathologist making diagnosis, normally they are standalone SAMDs or software as a medical device. So the intended user statement FDA normally required to include the already authorized, for example, IC assays, the CDX assay, and also the specific scanner viewing software, and also the display monitor. And FDA may apply the special controls, which is under 21 CFR 864 3750. So to include certain information in the labeling and also in the pre-market application for the FDA to review. So I just listed the requirements here. You can look it up, the special controls for such devices. And typically, the regulatory pathway in the US, that is a 510K. So it's a class 2 device. And then the next use case, that is a cancer prognosis, which Tong mentioned previously. Basically, this prescription device can provide prognostic risk estimate, but very importantly, it is in patients with previously diagnosed cancer. So in other words, the patient already been diagnosed to have cancer, and this device is to provide a prognosis or prognostic risk for the patient. And FDA outlined their requirements in the special control. So literally, I think the description of the data sets that is used to train, tune, and test the algorithm need to be provided. And also the device performance data need to demonstrate appropriate precision and reproducibility. The clinical data need to demonstrate the clinical performance of the device for the intended use. And last use case I really want to bring your attention to, that is about the biomarker assessment by using the AI-enabled computational pathology, and then that is used in the companion diagnostic device. And in April this year, Rose was granted FDA breakthrough device designation for the first AI-driven companion diagnostic, which is still under investigation for the non-small cell lung cancer treatment. And for such devices, I just want to emphasize the algorithm will be used to assess the biomarker status of the patient, either biomarker positive or negative. So when the biomarker is negative for a particular patient, then the patient will be eligible for the drug treatment. And so in this case, FDA considered it's a class 3 device. And normally the PMA or pre-market approval pathway is needed. So FDA will need to understand how the device works. For example, what is the device configuration, what is the clinical workflow, and what is intended use. For the biomarker, sometimes the biomarker could be the traditional biomarker, but often cases when we are using the computational pathology, the novel biomarker could be generated. So whenever FDA considers a novel biomarker, then the sponsor is supposed to provide very clear biological rationale and also scientific evidence to support the use of biomarker. And for the pharma company where I'm with, and the co-development with the companion diagnostic partner is needed to enable the concurrent review and approval. So in other words, the PMA from the CDX partner will be submitted almost at the same time as the drug sponsor to submit either BLA or NDA to the FDA. And when we are developing the algorithm, the training data set, test data set need to be independently assigned, and also for the analytical and the clinical validation of this algorithm. And of course, for the biomarker development, it should be driven by the evidence. So coming back to the second section of my presentation, I will provide a few thoughts on the practical solutions. So first of all, I think, again, I think it's very important for the FDA to understand what is your device configuration. And as I mentioned, even for SAMD or software as a medical device, normally that should be used with other FDA already authorized devices. So therefore, I think if it is IIC-based device, then a device is normally worked with the standard, which is already approved, the IIC assay the scanner and also the monitor display. And then the device configuration for registration purpose may be different. So in the US and the EU, such devices could be are considered as one device with multiple components. As I mentioned, the IC, scanner, algorithm, and the monitor display need to be put together as a combination device, not to be confused with combination product. Combination device are a device with multiple components in the US. And then they need to be put together into one submission for one device. However, in China and Japan, those components may require individual device application and registration. Okay, and the workflow is also very important. That leads to the role of the pathologist. So normally the pathologist is going to make the final diagnosis. However, in certain devices, the role of the pathologist may change. They may not be able to disagree with the biomarker status generated from the algorithm. Then the role of the pathologist will be shifted from making the final diagnosis to only performing QC or quality control. Then FDA will look into the device intended use. And then the second practical solution, I think, is related to the biomarker. So this is a particular case of when a biomarker is a novel. Traditionally, I think most of the IC assays, which already been approved by the FDA, they are membrane-based, assessed by the pathologist manually, either under the microscope or using a computerized solution on the computer. So there's still manual interpretation of the stained images. However, when a biomarker is novel, for example, instead of membrane staining, instead of assessing the membrane staining, maybe other factors can be utilized. For example, in one of the cases, I think it's already in the public domain for AstraZeneca's novel biomarker for CHOP2 that is assessing is so-called normalized membrane ratio. So it's assessing relative expression of the target protein CHOP2 on the membrane relative to the cytoplasm. So in this case, it's a novel biomarker as considered by the FDA. Then FDA does require very strong biological rationale to support the use of novel biomarker. So when you're developing the novel biomarker, the evidence should come from different lines of activities coming from the basic research, the preclinical that is based on cell line, and then the clinical development in different phases. Of course, I think it is very important to make sure what are the hypothesis generation cohort, and then what are the validation cohort. So the clinical trial need to be planned very carefully so that you can generate evidence and then you can finally validate the device very properly. And the next solution, I think, is normally the challenges is facing for the pharma company and also for the DX company, how to work together, how to co-develop the solution. And then the goal is, of course, to have a concurrent review by the FDA and get FDA's approval at the same time. So therefore, in order to work together, now I think, well, at least from AstraZeneca perspective, we try to bring the FDA into the scope at early time so that FDA is our collaborator instead of just the health authority to scrutinize the results. So we want them to collaborate with us. Of course, that require a lot of alignment with the DX partner on the device design and feasibility, align on the software algorithm development, and also what data will be used to support the biomarker assessment, the biomarker development, and also final device validation. And what is a global filing strategy, not only in the US, but you also need to consider in the EU, China, and also Japan. So this really requires a lot of functions within the pharma company and also with the DX company to work very closely and together with the FDA. And over here, the slide that I used very often, I just want to show you that you need to put very, very clear planning before the data set are used for the algorithm development. So normally the algorithm development is divided into 3 phases: training, tuning, and test. So you need to have the different data sets collected and put together for those activities. And finally, for the whole device, you need to have a very clear plan for the use of data for the analytical validation and also for the clinical validation. So FDA does require clear separation, and independent cohorts are used for different activities. And this is, I think most people, you have seen this. This is a good machine learning practice for medical device development. That is the guiding principles issued by multiple health authorities back in 2021. So those 10 principles, I consider it is very important. So I think you really need to look into each one of them and then try to abide to those principles during your development. And of course, for the biomarker, the particular one is a novel biomarker. You need to ensure the very strong evidence is collected to support the development of the biomarker and also the validation of the biomarker. So FDA does have a very good guidance document that is a biomarker qualification program. In here, it does provide a guidance regarding what kind of evidence is needed to support the use of biomarker. And I think that's the end of my presentation. I hope we do have some time for a panel discussion. So I will stop sharing.
Yeah, thank you, Chen, for your insightful presentation. So now we are open the floor to the audience questions. Again, please type your questions into the Q&A box. Yeah, again, thank you both Dr. In and Dr. Cui for your wonderful presentation. Now let's start with a general panel discussion question. I will start. So how do you see the AI pathology influencing the patient selection and clinical trial design in the oncology field? This question is to both Dr. In and Dr. Cui.
Tong, maybe you can start, I can chime in.
Okay, yeah, all right. Yeah, this is a great question. So this is a very, very big question. It's hard to answer. So I would say for the clinical development, and so I can't, because this is educational, I can't really talk too much about our current, there's a trial, but I think in general potentially is before it was one biomarker as a strategic factors, right? So let's use EGFR or ALK as an example, right? So you use the regular biomarker as strategic or stratification factor to predict the patient's outcome or predict the patient the treatment response for certain drugs. And this is a kind of a traditional way to do the targeted therapy, but with the new AI-driven approach, and which will be, I would say, is a more accurately to predict, because if you see here, so going back to some of the drugs, and so the response rate high, but it's not high to, so like a 60% of a response rate or 70%. Even you carry that mutation, it's not 100%, right? So why? And because so with the traditional biomarker approach, it's only looking one biomarker. With the AI, it's kind of combining the multiple angles to look at your omics data plus your image data, and or even from your like a patient characteristics, right? So from the race, from the age, from the the others, the patient histories, a lot of things you need to kind of put it together. So I think this is one areas the AI definitely can help, it can support, which I think the manual process so cannot achieve that. So that's my 2 cents. Over to you, Chen.
Sure, thank you. I think just now I just introduced a pre-screening tool. I think for a biomarker of very low prevalence, let's say only 10%, then I think the pre-screening tool by using computational pathology can predict the mutation status. And then if the patient is of a high risk to harbor such mutation, genomic mutation, then the central test can kick in and then to really confirm the genomic alteration status, and then the patient can be enrolled according to the eligibility criteria. And I think in addition to that, I think, at least big pharmas, they already started a plan to screen to do the pre-screening by using other technology, for example, NGS. And then they can look at multiple genomic mutations at a time. And of course, when you are enrolling a patient, maybe you want to use a specific molecular test, which will be your future CDX. Then if the same molecular test was used, for example, the NGS test was used during the pre-screening step and also in the trial screening, then I think maybe the concordance will be very high. So that can be directly used to support the marketing application. However, if you're using a different molecular test, then you need to consider what the level of concordance it could be, and whether the bridging is needed. Yeah.
Thank you, Dr. Yin and Dr. Cui for your answers. And I got a question from the audience. So this one is AZ developing AI model, and how to acquire patient data? Since this is the most critical roadblock for many companies.
Yes, so maybe I can start, and Tong, maybe you can chime in. Okay.
Yeah.
So I think there are certain proprietary information I cannot share, but you are right. AZ has already publicly stated we are developing AI technologies for sure. And then how to collect the patient information. I think we have our clean ops to set up the clinical trials, and then the patient will still be enrolled through the investigators, enrollment clinicians. And of course, I think the most challenging part for AZ is to come up with the biomarker, and also come up with algorithm to assess the biomarker accurately and also precisely. And this is what AZ is trying to achieve together with the DX partners. And we have already accumulated a lot of experience in the past several years. And this is why I think we are putting ourselves as a very strategic leadership in this field. So we are very confident going forward, but I'm pretty sure other companies are doing similar initiatives. I really hope the computational pathology can be used more broadly in such applications to develop novel biomarker that can truly benefit the patient in the long
run. Tong, do you want to chime in?
Yeah, I think Chen already covered everything I want to say, and so maybe just kind of comment on that. Yeah, so some of the internal development definitely we're doing, and so if you're talking about the patient data, and then because just like Chen mentioned, during the clinical and improvement, so that is where we can collect some of the data from the clinical perspective, but we also work with some of the externals, right? So this is not a secret. So AstraZeneca has already announced some of the partnerships. So for example, Tempus, right? So those are announcements out there. So there's many of those are different, either AI or driven companies where we're collaborating with, or the data companies. And we also worked with some of the kind of cancer centers, right? So there's a lot of collaboration to bring different data, and also bring the different model, trying to tune what we can do for the outcome.
Yeah, thank you, Tom.
There's another question here. So convolution is a calculus concept. How to apply this concept to figure out the slide image analysis?
Yeah, so I can probably, yeah, so since this is my slide, one of my slides. So the convolution, so neural network definitely is used in the math, but I think that the main point is here is trying to simplify or extract feature from the high resolution image, because the high resolution is big. We're talking about 100 by 100,000 of the pixels. It's one big image, right? And so if you're just looking at the whole image itself, it takes much computer power to do so. And first is from the cost perspective. The second, it will be from the time consuming perspective. It's not practical. So they have to do something with the algorithm to simplify the process. The convolution is the way, one of the ways to simplify and to extract the feature from the whole slice, and then still keep or contain the key elements. It's not losing all the features, right? So that is overall the concept.
Okay, thank you, Dr. In. So another question here is how digital pathology can help early cancer detection, and how reliable are the biomarker derived from the digital pathology?
Okay, so maybe I can correct on that. I think for early cancer diagnosis, I think is very important. I think most people know, well, for cancer, if it can be diagnosed early, maybe the survival will be more promising. So that is for the cancer treatment. Of course, I think most of the patients, whenever they have already gone through the pathology procedures, maybe very likely there are going to be lesions suspect to be cancer. Of course, I think there are pre-screening tools to do the well. There are screening devices for example, Cologuard, to detect maybe the possibility for the colorectal cancer, but that is not computational pathology based. So I think early detection, whenever patient have a sample, we want to ensure the algorithm can detect the small lesions which are likely to be missed by the pathologist, so that the cancer can be detected even though the lesion is very small. So hopefully that's the way the pathologist can work better. And then for a certain biomarker, what I can envision is by looking at the H&E stain slide, and then by looking at the morphology, even though there's no cancer, but maybe the cancer during the development can already have the pre-cancer tissue, maybe will already have certain morphological changes that can be caught by the AI algorithm. Therefore, maybe the AI algorithm can make a prediction whether the patient may develop cancer, let's say in 5 or 10 years, and then make a prediction over there. And then the patient can go through more frequent screening devices test, and then to ensure the cancer is not missed. And then if there are certain treatment options for early stage cancers, maybe they can consider to talk to their doctor to see whether they can apply it.
Thank you, Dr. C. So here there's another question. So how do you make sure the broad applications that can be implemented in not so rich hospitals or clinics down the road? Pathology lab results themselves vary drastically from lab to lab in terms of quality. I think this question is about the bias reproducibility.
Yeah. So I can share maybe first, is because that's the part of our job too, is to make sure first is the cost, the second it will be the quality, because one of our responsibility is we needed to cross check the different labs, different methodology. They all kind of provide equally high quality the data can predict for either screening or diagnosis that purpose, right? So, one way to doing that is EQA. I don't know you heard about the EQA, is external quality control, that kind of assessment. So because AI is fairly new, is because it's still evolving. There is not kind of like some of the traditional biomarkers you can easily to implement cross-check on their qualities, but there's still a certain way we can do, and so kind of a check is hospital by hospital. First is the variability, and the second is scanner by scanner, right? Or staining by staining, or even different algorithm by algorithms, right? So how those variables can play into that is something we are working on. Definitely, that is a very good question. So I think a lot of people are asking this kind of question, and this is definitely one of what we're doing.
Yeah, so maybe I just want to add that FDA normally they will require the sponsor to provide the training data set that may be representative of the real clinical use. For example, for Page prostate, FDA did ask Page to provide the training tuning test cohort, and then to see whether the slides from different labs around the world are used for training. So you can imagine different labs may have different staining protocols, as Tong mentioned, and also different scanner to be used. So the algorithm, FDA want to ensure the algorithm is already exposed to many different conditions from different labs, and then still can generate reliable results. So when you are developing such algorithm, you need to take this into consideration to ensure the algorithm is robust enough to deal with those different situations. And on the other hand, I think the SAMD, for example, for HER2 diagnosis, can really help the pathologist to try to eliminate, if not eliminate, try to lower the possibility to have the variability from different labs, and also the variability from different pathologists. So I think the computational pathology does have a very good opportunity to ensure consistent results and also accurate results is rendered to the patients.
Great, thank you. So here is a pretty general question. So biomarker was very useful for targeted therapy. However, since immunotherapy started, the biomarker study and testing were changed. What's your vision for biomarker study and the CDX in ADC, CAR-T, bispecific antibody, and even mRNA vaccine, mRNA therapy? Yeah. Do you try?
I think I already touched on this, there's one slide talking about different technologies, right, in a different area. I think, one of the areas we talk about, I think, Cheng also mentioned, is one of the trials in the trial 2, which is data already published, a year ago on the World Lung, but this year the World Lung we also have additional data published. So that is one of the ADCs. So we definitely using the digital biomarker to drive the drug to or the selection, the patient selection for the best the patient outcome. And then so others like CAR-T or first I didn't work on that, and second I think that is probably something we cannot share, even we have that information. So that is under development. If we have, it's not going something we already published, so we cannot disclose anything unrelated to those areas.
So maybe I can just add a few things. ADC stands for antibody drug conjugate. So I think ADC or other therapies you just mentioned, there are certain common mechanisms. For example, for ADC, the drug is a targeted chemotherapy per se, right? So the drug need to first bind to the antigen on the cell membrane and then get internalized, get into the cytoplasm, and the payload is released, and then go to the either the nuclei and then to stop the DNA replication, or disrupt the microtube in the cytoplasm. So of course the payload works differently. The common mechanism over here is the target, how to assess the target where the drug can take effect. So I think this really opens a new way by using a relatively old technology, which is immunohistochemistry, and to couple that with the computational pathology to really assess quantitatively how to measure such biomarker, and then by applying cutoff, it is converted into biomarker positive, negative, and then direct the patient treatment. So to me, I think computational pathology is really opening a door to the new world, to multiple novel therapies. So I think I'm really happy to see not only AZ, but also other companies are developing those therapies and that can benefit the patient in the long run.
Thank you,
Sure.
both speakers for your answer to this question. So now we have the last question that is about regulatory to Dr. Cui. So AI-enabled devices will need to have a clear plan for the use of data to train, tune, and test the AI model to validate the device. One question would be, what are the FDA's expectations on the data used for AI model and the device development?
Well, I think as a pharma company, I think you need to provide the clinical outcome data so that you can identify the biomarker. But developing a biomarker, I think it is not so easy work at all, particularly when you are coupling with the novel technologies. You need to consider the AI-based or AI-enabled algorithm, how you want to develop it. And then on top of that, you want to ensure you have the clinical data to train the algorithm to make a determination of biomarker. So as you can see, there are just so many data sets you need to consider and also plan ahead of time. This will take years and also very, very well-planned pathway going forward. So starting from the algorithm, you need to have a clear separation of cohorts for training, tuning, and test the data set. FDA has already provided very clear guidance on the requirements of those data sets. But normally, they need to be independent from each other on the cohort level. And then if it's not on the cohort level, then they at least need to be independent on the patient level. In other words, the data from the same patients are now used in both training and testing. And also, when you are testing the model, you cannot use the information you obtained from the test to inform your algorithm design. If you are using information from test to inform a new design of your algorithm, then the test data set is becoming a training data set from the FDA perspective. Then the FDA will ask you to provide another test data set to really assess the performance of the algorithm. And coming back to the biomarker development, it has to be assessed by associating with the clinical outcome. Then you have to also have a good separation of training cohort versus validation cohort. So when you are doing the training, you need to find the feature and also cutoff. You need to ensure you have sufficient number of patients in order to really identify what the cutoff would be. So a very good plan needs to be in place. And also you need to talk to the FDA to ensure your plan is a good, sound plan before you carry out. I think that will be the best approach going forward. Talking to the FDA early and often will truly benefit your development journey.
Yeah, great. All right. Okay, thank you. And last, thank you, Dr. Tong, Dr. Yin, and Dr. Cui for your wonderful presentation and the panel discussion tonight. So, Yinxi, I will let you to conclude. Thank you.
Thank you. Well, that concludes today's webinar. And on behalf of the CBA USA and the Clinical Translational Medicine Study Group, I'd like to extend our sincere thanks to our distinguished speakers for sharing their expertise, and also to our audience for your active participation and engagement. We truly appreciate it. And before we wrap up, I'm pleased to share that our next webinar will take place in just 2 weeks. On Saturday, October 11th at 8 p.m., we'll be focusing on a very timely and exciting topic: current challenges and opportunities of next-generation antibody drug conjugates. So we look forward to another stimulating discussion, and we hope you will join us. And thank you again for being with us today. Until next time, bye.
Thank you.
Thank you.
Thank
Bye.